Welcome to the deep dive. Today, we're taking a journey into the really intricate world of neurotransmitters.
That's right. The brain's chemical language.
Exactly. And we're using a core text on neurotransmission as our guide, sort of our map for this. And for you, the learner, our mission here is to really shed light on how these tiny chemical messengers manage, well, pretty much everything. Thoughts, feelings, movement.
We're aiming for clarity, those aha moments, focusing on the key players without, drowning in technical terms.
Right. Let's avoid the jargon, see. Okay, so big picture first. What are neurotransmitters fundamentally?
Okay, think of your brain like a huge network, like roads connecting those are the neurons.
Got it.
Neurotransmitters are the messages, the chemical signals that travel between these neurons. They trigger activity in the neuron that receives the message.
So they're the core communication units.
Precisely.
And it's not just one type of message, is it? There's variety.
Oh, absolutely. The text we're looking at groups them into four main categories. Monoamines, sometimes called biogenic amines.
Okay.
Amino acids, peptides, and then cholinergics. Acetylcholine is the classic example of a cholinergic one.
And the text seems to immediately highlight monoamines like dopamine, norepinephrine, serotonin, and also acetylcholine. Why are these ones so central, especially for neuropsychiatric conditions?
Well, it's because these specific chemicals are deeply involved in regulating really core functions. Things like our mood, our focus, motivation, feelings of pleasure, reward, that kind of thing.
Ah, the big stuff.
Yeah, the big stuff. So when these systems aren't working quite right, it can really impact mental health. That's why they're so key for understanding and treating conditions like depression, anxiety, schizophrenia.
Okay, that makes sense. So these messages are constantly being sent, but how does the brain stop them? They can't just keep firing forever, right?
No, absolutely not. That would be chaos. You need pauses, just like in a conversation. Signals need to be switched off. And the text mentions two main ways the brain does this. Two main deactivation methods.
Okay, lay them on us. How does the brain hit the brakes?
So the first way is super fast, happens right there at the synapse, the junction point. Think acetylcholine again. There's an enzyme, acetylcholinesterase, which acts like a tiny Pac-Man, just gobbling up the acetylcholine almost immediately after it hits its receptor, whether it's a nicotinic or muscarinic type.
Wow, okay, instant cleanup proof, very efficient.
Very. Clears the stage quickly for the next signal.
So what's the second method?
The second method is called reuptake. After the neurotransmitter does its job, binds to the receptor on the receiving neuron, it pops off.
Okay.
And then it gets actively pulled back into the neuron that sent it out in the first place, the presynaptic neuron.
So the sender takes its message back, like recycling.
Exactly like recycling. In this reuptake process, it's a major target for a lot of medications we use in psychiatry.
Ah, I see.
Once it's back inside, it can either be repackaged into those little storage bubbles, vesicles, ready to be sent out again. Or, if it's not needed right away, there are enzymes inside that cell that can break it down.
Right, right. And the text mentions monoamine oxidase, MAO. That's the enzyme that deals with norepinephrine, dopamine, and serotonin after they get taken back up.
That's the one, a really key enzyme. And, it raises the question, why doesn't MAO just destroy them before they get released?
Good point.
The answer is those vesicles. Before release, the neurotransmitters are protected inside these little membrane sacs. They're like tiny armored bubbles, shielding them from enzymes like MAO.
So they're safe inside their transport until launch time.
Got it.
Okay, so this brings us to the interaction, the dance, between the neurons, the neurotransmitters, and the receptors. Sounds complex.
It really is. And the crucial thing, especially thinking about treatments, is that most psychotropic drugs work by influencing the concentration of these key neurotransmitters. We're talking about dopamine, acetylcholine, norepinephrine, serotonin, histamine, GABA, glutamate, right there in the synaptic gap.
Okay, so how do they actually do that? What are the main strategies these drugs use to tweak that chemical balance?
Well, our source lays out a few main mechanisms. Some drugs are receptor antagonists.
Antagonists block things.
Exactly. Think of them as roadblocks. They fit into the receptor, but they don't activate it. They just sit there, blocking the actual neurotransmitter from binding and sending its signal.
Okay, so they stop the message. What else?
Then you have receptor agonists. These are more like master keys or mimics. They bind to the receptor and activate it, just like the natural neurotransmitter would, or sometimes even more strongly. So they boost the signal.
So antagonist is the brake, agonist is the accelerator. Makes sense. Other ways.
Another big one is interfering with that reuptake process we just talked about.
Ah, the recycling.
Right. Drugs can block the transporter molecules that pull the neurotransmitter back into the sending neuron.
So more neurotransmitter stays in the gap for longer.
Precisely. It hangs around in the synapse, giving it more time to interact with the receptors on the other side. Keeps the communication line open longer, so to speak.
Okay. What else is in the toolkit?
Some drugs can actually encourage the presynaptic neuron to release more neurotransmitters into the synapse, kind of like turning up the faucet.
Flooding the channel a bit.
Yeah. And finally, some drugs inhibit those enzymes we mentioned, like MAO, the ones that break down neurotransmitters after reuptake, or even sometimes in the synapse itself.
Like the acetylcholinesterase example earlier.
Exactly. By slowing down the breakdown, you also end up with more neurotransmitters available in the synapse.
Okay, that gives us a good framework. So let's dive into some of these key players. Dopamine first. What's its main job description?
Dopamine's a real multitasker. It's vital for movement, for integrating our emotions and thoughts, plays a big role in decision-making.
And reward, right. We hear about that a lot.
Absolutely. It's central to the brain's reward and pleasure circuits. Plus, it has bodily effects too, like stimulating the heart, increasing blood flow.
And clinically, when dopamine levels are off.
Well, the text points out that high levels are often linked with things like psychosis and mania, while low levels are associated with Parkinson's disease because of the movement role, and also depression.
Okay. The text mentions cocaine interfering with dopamine reuptake. How does that work?
Right. Cocaine basically jams the dopamine transporter, that reuptake pump. So dopamine can't get cleared out of the synapse. It builds up, massively overstimulating those reward pathways. That's where the intense high and the addiction potential come from.
Wow. And what about the dopamine hypothesis of schizophrenia? That sounds important.
Yeah, it's been a really influential idea. It came partly from observing that drugs which increase dopamine, like amphetamines, can sometimes trigger psychosis-like symptoms. And conversely, many effective antipsychotic drugs, like haloperidol back in the day, work by blocking dopamine receptors.
So it seemed like dopamine was the main culprit.
It pointed strongly in that direction, yeah. Well, now we understand schizophrenia is more complex, involving glutamate and other systems too. But dopamine is definitely still a key piece of the puzzle.
Okay. Let's shift to norepinephrine, or NE. What are its main gigs?
Norepinephrine is big on mood, attention, and arousal, keeping us alert. And it's a major player in the sympathetic nervous system.
The fight or flight response.
That's the one. Get you ready for action in stressful situations.
So some overlap with dopamine on mood, but different clinical links.
Right. With norepinephrine, increased levels tend to be associated with mania, anxiety states, maybe psychosis, whereas decreased levels are more linked to depression, feeling low energy, sedation.
The text mentions different norepinephrine receptors, alpha-1, alpha-2, beta-1, beta-2. Is it important to know the difference?
It definitely is, especially for understanding drug effects and side effects. Think of them as different docking stations that trigger different responses when NE binds.
Okay.
For example, the text mentions prazosin, used for PTSD nightmares. It works by blocking alpha-1 receptors. But interestingly, many older antipsychotics also block those same alpha-1 receptors. And that can cause side effects like dizziness from blood vessels dilating, low blood pressure, even issues with ejaculation sometimes.
So knowing the receptor subtype helps predict those unwanted effects.
Exactly. It explains both how a drug helps and what problems it might cause.
Very useful. Okay, let's talk serotonin, 5-HT, the one everyone associates with antidepressants. What's its range of functions?
Serotonin's got a huge portfolio. Mood regulation is the famous one, but also sleep, appetite, pain perception, aggression, libido, even hormonal activity. It's involved in a ton of stuff.
Wow. So what are the key clinical connections for serotonin?
Well, increased serotonin activity can be linked to anxiety. And in extreme cases, you can get serotonin syndrome if levels get way too high, which can be serious restlessness, tremors, even high fever, seizures.
Well, sounds so bad.
Can be. On the flip side, low levels of serotonin function are strongly implicated in depression.
Right. Now, the text mentions research is shifting towards strengthening serotonergic signaling. What does that mean exactly? Is it different from just boosting levels?
Yeah, it's a bit more nuanced. The thinking is moving beyond just, pouring more serotonin into the synapse. It's about improving the whole system's efficiency, how well serotonin is released, how sensitive the receptors are, the effects inside the receiving cell.
Optimizing the pathway, not just turning up the volume.
That's a great way to put it.
So how do SSRIs, the selective serotonin reuptake inhibitors, fit in?
SSRIs work specifically on the serotonin transporter protein called SERT.
Okay.
They block SERT, preventing it from sucking serotonin back up into the sending neuron. This leaves more serotonin hanging out in the synapse, available to stimulate the postsynaptic receptors.
Amplifying the signal that way.
Right. And the text makes a good point about a potential side effect. Because SERT is also found on blood platelets, SSRIs can have a mild effect on clotting. So there's a slightly higher risk of bleeding if someone takes an SSRI, alongside other drugs that affect clotting, like NSAIDs, ibuprofen, naproxen, or aspirin, warfarin, those sorts of things.
Good to know. Important interaction. What about histamine? We usually think allergies, right? Not so much mental health.
True, it's famous for the inflammatory response. But in the brain, histamine is also involved in alertness, wakefulness.
Ah, keeping you awake.
Exactly. And it influences things like gastric acid secretion, too. But in psychiatry, the main relevance comes from drugs blocking histamine receptors, specifically the H1 type. Lots of older psychiatric meds do this.
And what happens when you block H1 receptors?
The two big side effects are sedation, feeling drowsy, and weight gain.
Ah, the common ones.
Yep. The sedation might sometimes be useful short-term, say for agitation, but the weight gain can be a real problem long-term for health.
Makes sense. Okay, next up, GABA. Gamma-aminobutyric acid, often called the main inhibitory neurotransmitter. What does inhibitory really mean here?
It basically means GABA's job is to calm things down, to reduce neuronal firing. It acts like the brain's brakes.
Okay, a brake system.
Yeah, it helps regulate anxiety, prevents seizures, that's its anticonvulsant property, and it helps muscles relax. The text also mentions it might play a role in pain perception, but cautions that too much GABA activity could potentially impair thinking or motor skills.
So it keeps things from getting too electrically excited in the brain?
That's a good way to think about it. Keeps things stable. Clinically, increasing GABA's effect generally reduces anxiety. That's why many anti-anxiety drugs target this system.
Right.
Conversely, decreased GABA function has been linked to mania, anxiety disorders, maybe even psychosis. Suggests GABA is crucial for maintaining that neural equilibrium.
And how do those anti-anxiety drugs usually work with GABA?
Many of them, like the benzodiazepines, Valium, Xanax, that class don't mimic GABA directly. Instead, they bind to the GABA receptor at a different spot and make it work better when GABA itself binds. They enhance GABA's natural braking effect.
Ah, they boost the brakes.
Exactly. And the text notes an interesting research direction, developing new antipsychotics that might tackle both dopamine hyperactivity and GABA hypoactivity in conditions like schizophrenia, trying to hit the problem from both sides.
That sounds promising, addressing multiple imbalances. Okay, let's flip to the other side. Glutamate, the main excitatory one.
That's right. If GABA is the brake, glutamate is the accelerator. It's the primary neurotransmitter for speeding up neuronal activity. Hugely important for learning, memory. Most brain functions rely on it. It acts mainly through NMDA and AMPA receptors.
NMDA and AMPA.
Yeah, AMPA receptors seem particularly crucial for the mechanisms underlying learning and memory formation.
And what happens when glutamate signaling goes wrong?
Well, the text highlights a couple of key issues. Too much stimulation of NMDA receptors can lead to something called excitotoxicity.
Sounds bad.
It is. Neurons get overstimulated to the point of damage or death. This is thought to play a role in neurodegenerative diseases like Alzheimer's. On the other hand, reduced activity at NMDA receptors has been linked to psychosis. And interestingly, boosting AMPA receptor activity seems linked to better cognitive performance, though again, too much can be harmful.
So it's all about balance. The text mentions the glutamate-GABA balance being critical, especially in psychosis.
Absolutely critical. You need that push-pull between excitation, glutamate, and inhibition, GABA, for the brain to function properly. The text mentions memantine, which blocks NMDA receptors somewhat, used in Alzheimer's partly to reduce that excitotoxicity.
Right.
It also mentions a newer drug, lumateperone, brand name Caplyta, which has a complex profile affecting serotonin, dopamine, and glutamate pathways.
Interesting.
And that whole interplay between AMPA and NMDA receptors is central to how newer, rapid-acting antidepressants like ketamine and related drugs are thought to work.
Very cutting edge. Okay, circling back to acetylcholine, ACH. We know it's involved in muscles. What else does it do in the brain?
Beyond muscle control, ACH is vital for things like learning, memory formation, attention, regulating arousal, and the sleep-wake cycle.
So cognitive functions too.
Big time. And it also modulates mood imbalances that are linked to both mania and depression and can affect sexual and aggressive behaviors. Plus, it runs the parasympathetic nervous system, the rest and digest side of things.
Quite a portfolio. What are the main clinical links for HM imbalances in the brain, besides the muscle stuff? Alzheimer's comes up again.
Yes, a significant drop in HM function is a key feature of Alzheimer's. It's also implicated in Huntington's disease and Parkinson's disease. Interestingly, some studies suggest increased HM activity might be linked to depression, and the text emphasizes that balance between HM and dopamine is really important.
Right, and those Alzheimer's drugs, the HM inhibitors like donepezil, how do they work again?
They block the enzyme acetylcholinesterase HA. That's the enzyme that normally breaks down acetylcholine in the synapse.
The Pac-Man enzyme we talked about earlier?
Exactly. By inhibiting that enzyme, these drugs increase the amount of HM available in the brain, which can help boost cognitive function, at least for a time, in people with Alzheimer's.
Okay. The text also brings up nicotine and nicotinic receptors. What's the connection there?
So nicotinic receptors are a specific subtype of acetylcholine receptor that, as the name suggests, nicotine binds to very effectively.
Ah.
The text mentions that people with schizophrenia or attention issues sometimes have differences in these nicotinic systems. There's a theory they might smoke using nicotine as a form of self-medication to try and normalize some cognitive or sensory processing issues.
Interesting theory.
Yeah, but obviously smoking is incredibly harmful. So researchers are looking for ways to develop drugs that target these nicotinic receptors more safely without all the addictive and dangerous aspects of tobacco.
Makes sense. Okay, finally, a quick look at peptides or neuromodulators. The text gives examples like substance P, somatostatin, neurotensin. How are these different from the neurotransmitters we've mostly discussed?
They're generally thought of as modulators rather than direct, fast-acting transmitters. They tend to fine-tune or adjust the overall activity of brain circuits, often influencing how neurons respond to the faster neurotransmitters, like glutamate or GABA. Their effects are usually slower to start, but longer lasting.
Like volume or tone controls, maybe?
That's not a bad analogy, yeah. They modulate the main signal.
And those specific examples, substance P?
Involved in memory, mood, anxiety, and definitely pain perception. Drugs that lock its receptors are being explored as potential antidepressants or anti-anxiety agents.
Somatostatin.
Levels seem altered in cognitive diseases. It's often decreased in Alzheimer's and some people with depression, but increased in Huntington's disease.
And neurotensin.
Research suggests it might have some natural antipsychotic-like effects. Levels have been found to be lower in the spinal fluid of some individuals with schizophrenia.
Okay, fascinating how many different chemical players there are. So we've covered the main messengers. Let's shift to how psychotropic drugs interact with all this. The text admits we don't fully understand exactly how many of them work, which is humbling.
It absolutely is. Shows the brain's incredible complexity. Science is always learning. So the text then gets into drug interactions, starting with pharmacokinetic ones. Remind us what those are about.
Pharmacokinetics. That's about what the body does to the drug, right?
Right.
Absorption, distribution, metabolism usually in the liver, and then elimination, how the drug moves through you.
Exactly. And these processes determine how much drug actually gets to your brain. The liver enzymes, particularly the cytochrome P450 system, CYP450, are huge players here. They break down lots of medications.
Okay.
Now, some drugs can inhibit these CYP450 enzymes. Think of it like slowing down the liver's processing line.
Which would make other drugs build up.
Precisely. It can increase the levels and potential toxicity of other drugs metabolized by the same enzyme. Conversely, some drugs are inducers. They speed up the enzyme activity.
So the other drug gets broken down faster, maybe becoming less effective.
You got it. Unless the dose is adjusted. And the text points out many newer antipsychotics rely heavily on liver metabolism, making them prone to these CYP450 interactions.
And this is where pharmacogenetic testing comes in, things like GeneSight.
Exactly. These tests look at your specific genes related to those CYP450 enzymes and sometimes neurotransmitter targets too. It can give clues about how quickly you're likely to metabolize certain drugs, helping doctors choose medications and doses that might work better for you personally, with fewer side effects. More personalized medicine.
Okay, so that's pharmacokinetics, one drug affecting another's level. What about pharmacodynamic interactions?
Pharmacodynamics is about what the drug does to the body, or in this case, the brain. It's about interactions at the target site, the receptors, the enzymes.
Ah, so drugs affecting the same system.
Right. Two drugs acting on similar receptors or pathways. They can have synergistic effects. The combined effect is more than just adding them together.
Yeah.
Or antagonistic effects. One drug cancels out or reduces the effect of the other.
Got an example.
The text gives a common one. Combining a benzodiazepine, which enhances GABA and causes sedation,
Right.
with an older antihistamine that also causes sedation by blocking histamine H1 receptors.
Yeah.
Put them together, and the sedative effect could be much, much stronger than either one alone, potentially dangerously so.
I see. So managing these interactions sounds incredibly important, especially for people on multiple medications like older adults often are.
Absolutely critical. It requires really careful review of everything a person is taking, understanding their individual health factors to keep treatment safe and effective.
Okay. Lastly, the text touches on antidepressants and the theories about depression. It makes a key point. Even the old monoamine hypothesis, the idea that depression is just low dopamine, norepinephrine, or serotonin, doesn't have solid proof.
Yeah, that's important. While drugs targeting those systems do often work, it's probably not as simple as just a chemical deficit. The monoamine receptor hypothesis is a bit more complex.
How so?
It suggests that maybe the problem isn't just low neurotransmitter levels, but that the receptors for those neurotransmitters become overly sensitive or increase in number, upregulate, as a way to compensate for low levels over time.
Ah.
And maybe antidepressants work by gradually correcting that receptor sensitivity, which could help explain why they often take weeks to really kick in. The brain needs time to adapt back.
Okay, that makes more sense than an instant fix. What about newer ideas?
Research now is looking more at downstream effects inside the cells, things like gene expression, cellular pathways. There's a lot of focus on BDNF, brain-derived neurotrophic factor.
BDNF.
That's like fertilizer for brain cells.
Yeah.
Helps them grow, survive, make connections. Some evidence suggests BDNF might be lower in depression, and maybe antidepressants work partly by boosting BDNF levels, promoting brain health and resilience.
So it's way more intricate than just low serotonin.
Oh, definitely. It's about networks, plasticity, cellular health. Much bigger, more dynamic picture that we're still figuring out.
Well, this has been a truly fascinating deep dive. So for you, the learner listening in, what's the big takeaway message here?
I think a core insight is just how fundamental these neurotransmitters are to everything our brain does, and how tightly linked they are to mental health. Understanding their roles, how they're regulated, how drugs influence them, even with the complexities and unknowns, gives us really crucial perspectives on both normal brain function and mental illness.
Absolutely. And maybe a final thought to leave you with. Considering this incredible complexity and the fact that everyone's neurochemistry and drug metabolism is slightly different, what does that imply for the future? How might we move towards more truly personalized approaches in mental health care? It really makes you think about where the research is heading and the potential for treatments that are far more targeted and hopefully more effective for each unique individual. Thanks for joining us on the Deep Dive.